Hi, so let's start with the microbiology, very small topic, mycology. Mycology is the study of fungus. Okay, so fungi are eukaryotic, obligate, facultative aerobes. They are aerobes and they are natural habitant of soil, water, and organic debris. Soil, water, and organic debris. Okay, cell wall contain chitin, mannan, and other polysaccharides. Cell wall contain chitin, mannan, and other polysaccharides. Morphological classification of fungi are of four types: yeast-like fungus, molds, and dimorphic fungi. Yeast-like fungus, molds, and dimorphic fungi. Yeast, round to oval unicellular fungi. These are round to oval unicellular fungi reproduced by budding. Okay, like this, and form creamy mucoid colonies on culture. Example, cryptococcus neoformans. Cryptococcus neoformans reproduce by budding and form creamy mucoid colonies on culture. Okay, yeast like fungals. Yeast like fungals go partly by like yeast, like budding, and partly by chains of budding cells joined end to end, like this, pseudo hyphae. Chains of budding cells joined end to end, pseudo hyphae, like this. Example is Candida albicans, which is the most important example to us. Then molds. Molds grow as branching filaments. Hyphae, they are aseptate or septate hyphae. Hyphae are of 2 to 10 micrometer in width, and a tangled mass of growth is called mycelium. In culture, above surface aerial mycelium. In culture medium, inside the culture medium it is vegetative, and above the surface it is aerial mycelium. Okay, and example are aspergillosis, rhizopus, penicillin, and dermatophytes. Okay, these all form hyphae, grow as branching filaments. Okay, then dimorphic fungi in host tissue exist as yeast. Dimorphic fungi, it's dimorphic means have two morphology like yeast, or in culture it is like hyphae. Okay, in host tissue like inside us it will exist as yeast, and in culture at 22 to 25 degrees celsius it exists as hyphae, culture or soil hyphae. Most of the systemic infections, okay, most of the systemic infections are dimorphic fungi. Now, another things, some important points about fungi are they have rigid cell wall which contains mannose, chitin, and other polysaccharides. Cytoplasmic membrane contain sterols. Cytoplasmic membrane contains sterol. Cytoplasm are true have two true nucleus, nuclear membrane, mitochondria, and endoplasmic reticulum. They are uni or multicellular in nature. Okay. Then examples of most of the systemic infections fungi, dimorphic fungi are blastomyces, coccidioides, immitis, and histoplasmosis. So coccidioides, blastomycosis, myces, and histoplasmosis. And then lab diagnosis of fungal diseases. Microscopy. In microscopy, we have four things: KOH preparation, potassium hydroxide, 10% potassium hydroxide, then potassium hydroxide with calcofluor white, which which is seen as fluorescent bright green color. Okay, in calcofluor white, and then it's like fluorescent green. And then gram positive yeast candida in gram staining, and then India in preparation, capsulated yeast, example Cryptococcus neoformans in CSF. Okay, India in preparation, cap for capsule of the Cryptococcus neoformans. And then second is culture. Culture are of two types: SDS, Sabarods, dextrose, agar medium, and Sabarods with antibiotics. Antibiotics contain chloramphenicol, not for bacteria, cycloheximide containing fungi. Okay, then tissue sections have by which we stain are periodic acid shift and methylamine silver stain. And then serology can also be performed. PPT test with carbohydrate extract of candida. Serology in precipitation test with carbohydrate extract of candida. Then fifth is ELISA can also be performed for antigen detection. ELISA can also be performed for antigen detection, skin test, indicator of cell mediated. Okay, these are for candidas. For common fungus lab diagnosis are these three only: tissue sections in which two stains are used: PAS and methylamine silver. In culture, several texture SDA or SDA with antibiotics, and in microscopy, these four. Okay, and then candida. Okay, most commonly seen in 80 to 90% of population, candida albicans only. Then it is an we all know it is an opportunistic infection seen in immunocompromised or terminal illness. Infection of skin infection of skin, mucosa, and internal organs. Other species are Tropicalis crucei, glabrata, and Vishwanathi. Predisposing factors are immunodeficiency, malignancy, prolonged antibiotic intakes, suppression drugs, and IV catheters. Okay. Then clinically, mucocutaneous lesions are seen in oral cavity, vaginitis, conjunctivitis. Okay, and in skin and nail manifestations as axilla, seen in axilla, groin, perineum, and submammary folds, which are the folds beneath the breast. Okay, then nails we have polyonchia, and in systemic candidiasis, UTI, urinary tract infection, intestine diarrhea, in intestine, septicemia, endocarditis, meningitis. Okay, meningitis. In MCQ, we all get meningitis, and I thought before that candida is not a cause of meningitis, but it is. Okay, endocarditis, intestinal disease like diarrhea, UTI, septicemia, candida are the cause of all these. Okay, so please read this again because we get options like these. Okay, oral cavity. Okay, mucocutaneous lesions are in oral cavity, vaginitis, conjunctivitis, or keratitis also. And then skin and nail manifestation, perionychia, submammillary folds, perineum, axilla, groin. And in systemic diseases, UTI, intestine, diarrhea, endocarditis, in heart also, and in septicemia also. Then fourth, chronic mucocutaneous candidiasis. And then lab diagnosis, in microscopy we do direct KOH or gram staining. In culture we do sabrostextrose agar cream colored colonies. To differentiate candida from other species, we do gram tube test and chlamydospore formation. Okay, in gram tube test we get ability to form germ tube within 2 hours when incubated in human serum at 37 degrees Celsius. Incubated in human serum. Ability to form germ tube like this. This is the germ tube and this is the yeast cell and candida cell. And then ability to form germ tube within 2 hours when incubated in human serum at 37 degrees Celsius. This is called as Reynolds-Brodé phenomena. This is called as Reynolds-Brodé phenomena. This I got this question and I I I don't do it correctly because I don't remember it. So please remember this. Reynolds-Brodé phenomena is seen in candidiasis, and it is a test to differentiate candida from other species. Okay, and gram tube test, in which it forms germ tube in 2 hours at in human serum at 37 degrees Celsius. Okay, and then we have chlamydospore formation in nutritionally deficient media. Example, it forms chlamydospore. Okay, examples: cornmeal agar at 20 degrees Celsius. And then fourth is serology, PPT test for carbohydrate extract of Candida. Okay, this is for candidiasis. Serology, precipitation test for with carbohydrate extract of Candida. And then fifth is ELISA, antigen detection for Candida. Skin test, indicator of cell mediated immunity, which is universally positive test. Then that's all about Candida albicans. And then in microbiology, that's it, only one page you got. In this, you have to remember that albicans, you all know that it's the most common species of, Candida. And then Tropicalis, Crozet, Glabrata, and Vishwanathi we can get in the options. And then Chlamydospore is seen at end of pseudohyphae. Chlamydospore seen at end of pseudohyphae. Okay. Then these lesions we get, okay, clinically in Canada, and this is the lab diagnosis, in which Reynolds Brodie phenomena is the most important. KOH and SDA are mostly used, or also it is gram positive, so gram staining can also be done. Okay, then Cryptococcus. Okay, in this, Cryptococcus neoformans, microscopically encapsulated yeast. It is an encapsulated yeast. India incorporation or nigrosin stain we can do like this. It is seen, okay, black direct microscopy. Indian preparation or nigrosin stain. Okay, soil saprophytes in soil, feces of pigeons. In pigeons, it is present. Cryptococcus neoformans is present in the feces of pigeons, but it does not infect pigeons because they have increased body temperature and gets mixed in soil saprophyte, and does not infect birds because increased body temperature. Only deep mycosis in India, mostly seen in immunocompromised individuals, majority asymptomatic. The majority are asymptomatic in pulmonary cryptococcus. Majority are asymptomatic in pulmonary cryptococcus. Mild pneumonia is seen. In pulmonary cryptococcus, mild pneumonia is seen. In HIV, cryptococcus meningitis or hematogenous, okay, spread. In pulmonary cryptococcus, mild pneumonia is seen, and in HIV patients, cryptococcus meningitis because of hematogenous spread is seen. Okay, and in HIV, infection acquired by inhalation of dust containing yeast cells. Okay, it is also gram positive. Then lab diagnosis. First is microscopy, in which 5 to 15 micron in diameter capsule. Oh, sorry, not capsule, but the cells are seen, okay, of cryptococcus. The fungus are seen. Spherical budding, okay, cells. Spherical budding cells have prominent polysaccharide, prominent polysaccharide capsule. Okay, if the capsule in capsule question comes, then it is CC, Cryptococcus neoformans. Okay, mostly in fungus. Gram positive yeast, and then India ink preparation in nigrostain, like this. It is seen like this in India ink preparation or nigrostain. Okay, then majority cases are asymptomatic. And then second is culture media we use is SDA, incubated at 37 degrees Celsius, smooth cream colony seen. And then differential media is Niger seed, bird seed agar or Niger seed. Okay, in this black brown color, colonies are seen. Specimen CSF sputum, pus, brain tissue, okay, are the specimens that is collected. Differential medium is Niger seed brain, birdseed agar. Birdseed we can remember like it is because of pigeon. So birdseed agar or Niger seed agar, which is seen as black brown colonies. And then for detection of capsular polysaccharide antigen, latex agglutination test is used, divided into A to D, four types A to D, divided into what capsular polysaccharide antigen for detection of capsular polysaccharide antigen, divided into A to D, four types, latex agglutination test. Okay, then next fungus is aspergillosis. Oh, sorry, not aspergillosis. It's rhizosporidiosis, associated with chronic granulomatous disease. Granulomatous disease seen in nose, mouth, and eye. Friable polyps in nose, mouth, and eye. Friable polyps are seen. Characteristic. Then life threatening. It is life threatening. Increased risk in COVID-19 disease by organism is Rhinosporidium sibrae. Rhinosporidium sibrae. Okay. The infection is of unknown origin and seen in male segment of H2O aquatic life. Okay. Just the thing to remember. Male stagnant H2O in males which are swim in stagnant water or aquatic life. Okay, then lab diagnosis has not been cultured. In this culture, we don't perform. We do microscopy, which is diagnostic hallmark of sporangia on H&E, large number of endospores with the sporangia. Then pneumocystic cerevisiae, uh, pneumocystic carve seen in rats. Karen. Pneumocystis carinum is seen in rats, and this is seen in cervica, seen in humans. Okay. Most common cause of pneumonia in HIV patients is because of pneumocystis cerevisiae. As overall, most common cause of fungal disease in HIV is Candida, but most common cause of pneumonia in HIV patients or okay, it's pneumocystic cerevisiae. Opportunistic infection. It is an opportunistic infection. Route of spread is respiratory route. Then infective stage is cyst. Then sample for diagnosis sputum, bronchoalveolar lavage, bronchoalveolar lavage or lung biopsy. And then treatment we do is we can give methoxazole, which is a combination of trimethoprim and sulfamethoxazole. Okay, then direct demonstration of trophozoite form. Direct demonstration of trophozoite form is Gymnasia toluidine blue or methylamine silver. Methylamine silver, direct demonstration of, and that's now our fungal infections in microbiology for MDS is completed.